Thank you all for coming to tonight's event, Deciphering DNA. I'd like to start by acknowledging the Kaurna people, the original custodians of the Adelaide Plains, and the land on which the University of Adelaide's campuses here at North Terrace, at Waite and Roseworthy are all built. My name is Hayley Parkinson and I'm undertaking my PhD in the School of Biomedicine here at the University of Adelaide, and I'm going to be your MC for tonight. More than 500,000 Australians have a neurodevelopmental disorder, from intellectual disabilities and epilepsy to movement and behavioural conditions. These disorders are usually of genetic origin, yet only half of individuals diagnosed also receive a genetic diagnosis. This limits their access to therapies, customised care, and empowered family planning decisions. Today, you're going to hear from Dr. Lachlan Jolly, who is working to change this. Lachlan leads the Neurobiology Research Group in the Robinson Research Institute and the School of Biomedicine here at the University of Adelaide. He is also the past president of the Human Genetics Society of Australasia for the SA branch. Please join me now in welcoming Lachlan.
Cool. Thank you, Hayley, and thank you, Charlotte, and thank you all for being here today. I dedicated my life to trying to research for the benefit of human health, and in particular those with neurodevelopmental disorders. And so it's always a privilege to be able to speak to the public because, in a sense, I'm funded by the government. You guys pay taxes to the government, so I consider you my clients. And if I can inspire you to support research like what I'm doing, hopefully we can all pull together to make this world a better place for, in particular, children who are born with a genetic defect that really limits them reaching their full potential. And I'm sure as all parents in the room, or brothers or aunties would hope, when you have a new child join your family, you just want them to have the best possible chance they can to be the best person they can be. And when you have a born with a genetic defect, a genetic variant that limits that, that could be challenging to deal with. So I just want to also acknowledge that today we're meeting on traditional Kaurna people country, the Adelaide Plains. It's a place I've called my home for most of my adult life. I moved to Adelaide when I was in my late teens, and this particular lecture theatre is a place I hold strong. It's the first time I ever did a lecture in biology, and here I am some 25 years later providing this lecture. So it's a bit of a full circle for me. But this land is a place I love, whether I'm living at the beach or now where I'm living in Belair Hills. It's just a magical place for me. And as I built all my memories here with my family and build my own connection with the lands and waters, you can't help but pay your respect to the beauty that we're living in here in Adelaide. And for that, I pay my heartfelt respect to the for the elders past, present and emerging that have maintained the cultural importance, the heritage and the beauty of this land. So thank you. So we're here to talk about today deciphering DNA. This is really important, an emerging era that we live in, and that is an era of high access to genetic information and genetic understanding of the genetic basis of human health. DNA was not discovered that too long ago. As I can see some grey hair in the audience, it was probably discovered during your lifetime. When I joined, it was a new, dazzling thing, 20 years old since we discovered DNA and its importance in inheritance. We're in a stage now where we can sequence people's genomes really rapidly and get a good understanding right from the early stages of life, what their life trajectory might look like. And it's really important for those living with what we classify as rare disease. So a rare disease is just classified as someone with a disorder that has a frequency of less than 2,000 people in the population. And despite their name being called rare disease, collectively they're quite common. There are over 6,000 individual rare diseases, and they affect somewhere around 5% of the population. That's 300 million people. That would be by population the third biggest nation in the world. Okay. Now, the majority of rare diseases arise actually during childhood, 70% of them, and another around 70% of these disorders are of genetic basis, meaning that these are children from day one that are born with that inherited a change in their DNA sequence, as I mentioned before, that's going to limit their quality of life. At the other end of the spectrum, only 10% of rare diseases, in particular rare genetic diseases, actually have a therapy. So whilst at the moment we can diagnose individuals based on their DNA sequence, how that translates to improvement in their clinical care still remains a very large challenge, but a very big opportunity at the same time. So what can a genetic diagnosis give you? Well, it can give you the appropriate care. Now, it might not give you the tailored care to completely be the magic bullet that solves your ongoing issues, but it can be appropriate care. It can tell you what not to treat with. You can imagine a child with an epilepsy. An epilepsy you can treat with many different drugs, but depending on the genetic mutation that underpins that epilepsy, one drug might be very deleterious, the other drug might be very efficacious. So there is important decisions to be made, even if it's not cure, about appropriate care and access to therapies. If you have a child that has an early diagnosis, that might then be able to teach you about their likely lifehood trajectory. Will the child eventually, like a child with maybe a delayed speech or movement disorder, will they grow out of it? Will it get worse? Is it progressive? Can we expect other life issues to arise? Will they be susceptible to infections? This sort of information is really important. Access to clinical trials. So whilst only 10% of rare diseases have a therapy, about another 14,000 clinical trials are currently underway that are targeting about another 1,500 of these rare diseases. But to get access to this new and emerging fantastic opportunity for a clinical trial that might be the difference between life and death with your child, potentially, you need that. First of all, you need that genetic diagnosis. That way they can triage the right individuals into the right trials to get the best outcomes. But it's all downstream of a clinical diagnosis. And last thing is family planning. So all of what I've just talked about is really related to the affected individual. How can we help this child? But these diagnoses help whole families. You can imagine I'm the father of a child that has an intellectual disability, and there's a brother on my dad's side that also has an intellectual disability, and I've got a sister that wants to start a family, but she's cautious. She's less clear whether this slight family history may lead to her having a child in which may not reach its full potential. And usually what a genetic diagnosis can do is just empower those family planning decisions. Okay, my sister doesn't have the particular variant that's causing the disorder, and boom, she has a whole family of her own. Or potentially she knows she has the variant, but she can manage and make her decisions around whether to start a family and how to handle those decisions moving forward. And the last thing I have is the community, because this is really important for rare diseases. Like I mentioned, there are 6,000 rare diseases. It's unlikely that if you have one, say in Adelaide, it's very unlikely that there'll be another family going through the same problem in Adelaide as well. And in fact, there might only be a handful of families worldwide, maybe a few hundred families worldwide. But we're living in an age now where, speaking with someone on the other side of the world is not a challenge. We just jump on Facebook, we start a group, we can bring people together. And this is really important because if you do have a rare disease and you're one of, say, 100 people, who are the experts in that rare disease? It's the parents of that child, right? Because they have the lived experience of what therapies have worked, what therapies have not worked, what drugs have had an impact, what shouldn't I do, what situations should I involve. So the parents actually end up being the experts of these rare disorders, and putting them together as a community is really important. You can have a mother of a child who's 18 years old, who's been through all the trauma of the tweens and the drugs, the therapies which worked and didn't work, speaking to another mother whose child, 1 year old's just been diagnosed, telling her what to expect and what maybe can work best for her in her situation. So the community is super important for rare diseases. Now, so the diagnosis is really important, okay? And currently only about 50% of the people with a suspected genetic disorder get a diagnosis. And on the other end of the spectrum, I've mentioned to you that only 10%, or even less than 10%, of those with a rare disorder can actually have access to a therapy that's going to make a difference. And that's underpinned by the fact that we simply don't know enough about the genes and what these genes do that are implicated in these disorders to actually be able to make a tailored therapy. A defect in gene X means I need to have drug Y. There are very few relationships like that, simply because we lack the fundamental knowledge about gene X in the first instance. And there are a lot of genes in your genome, about 22,000 of them. About 3,000 of these are actually involved in neurodevelopmental disorders, and these genes are all involved in developing and making your brain work, the most complex organ in the body. You can understand that there's a very large task in front of us to unravel all of this. So neurodevelopmental disorders are classified essentially by a significant disruption during brain development. This can be a child that really just, when you first notice, is not meeting some of the early developmental milestones, like learning to walk and talk, maybe even creating eye contact, these sort of gestures and smiling, just a lack of maybe censoring of the environments or these key motor skills. And they it's a spectrum. We all have children, we all learn at different rates. There's no right or wrong, but I guess what I'm really talking about here is individuals that are really right at the end of the spectrum, that don't come out of these delays, but that go on to have problems with learning and memory and other neurological based processes throughout their life. So some names here which you maybe have heard of: autism. I'm sure everyone in the room is aware of autism. It's maybe not so much of a disorder, but neurodiversity. But some of these things like Rett syndrome, for example, was quite a severe disorder. And these individuals actually struggle to, struggle to contribute to society in a normal way. They need a lot of help and support. So you probably wouldn't see someone with Rett syndrome walking across the street or catching the bus with you. These people are in places where they can receive high levels of care to make their life safe and enjoyable as possible. Other things, so a lot of these rare disorders don't have names. And in fact, there's a whole group, patient advocates that support these umbrella conditions. They might be described more based on their symptoms. So you might have seizures or epilepsy as a symptom, or you might have intellectual disability as a symptom, but a lot of these syndromes just simply don't have names. Okay, and in Australia, they affect around about 1 in every 50 children. So that's 2% of the population. And that might seem like a really high number, but again, as I mentioned before, these individuals are not necessarily visible to the general population because they are in places where they can receive the acute care and treatment to keep them safe and keep them happy. So like I mentioned, these are not necessarily people that you come to deal with that often. Like I mentioned, I come from a small country town in Cleve, a couple of hundred people. And in that small country town, I can tell you that I had about 5 or 6 friends, young children with neurodevelopmental disorders. And why they're living in small country towns? Because it's safe. They don't have to worry about crossing the street, buying things. They're in a place where everyone knows everyone and everyone can keep them safe. And you'll find these people in these small country towns living their best life. Okay, so the causes. Now, I'm a geneticist, but not everything is genetic. Indeed, these neurodevelopmental disorders can arise during childhood or during pregnancy from environmental factors. And this is particularly relevant to probably somewhere like Africa where the level of healthcare is not that great. So you might have an infection during pregnancy, you may be malnourished during pregnancy, or the child may be malnourished, and this can actually affect your trajectory of neurodevelopment. But in somewhere like Australia, where the healthcare system is among the best in the world, and things like, where everyone's vaccinated, everyone's having a reasonable access to nutrition and diet, the major cause by far, by a long way, is a genetic cause. And these are simply things that we just cannot control. A lot of these genetic defects arise spontaneously in the eggs of the father or the mother. Sometimes they're passed through family generations and you can pick it as being genetic. But often with these really severe neurodevelopmental disorders, there isn't a family history because these children simply don't reproduce, right? So they often just arise what we call de novo for the first time ever. And we simply can't stop that from happening. There's no way of predicting that your child that you're making might carry a deleterious DNA variant that has arisen all of a sudden in one of your sperms or one of your eggs. Okay, so here is a more classical picture of a genetic inheritance. We have, this is a family tree where the squares represent males in the family and the circles represent females in the family. And towards the top here, you can see they come together and they have themselves 6 or 7 kids. These are probably baby boomers, right? And what we also have here is in colours, the individuals that are coloured in orange, individuals which we classified as having a clinical symptom of this neurodevelopmental disorder. So you might have an individual come into the clinic down here, and the first thing they ask is, what's your family history? Maybe he has an epilepsy. Oh yeah, my mother kind of had some seizures, and then my mother's father also had some sort of epilepsy, and my mother had a brother had an epilepsy, and they'll decide that we need to perhaps go and see whether there's a genetic test that can maybe underpin, find out the reason behind this epilepsy, and hopefully tailor a treatment, if not a therapy. Okay, so then it goes to, you take a blood sample. Every cell in the body has the same type of DNA in it. That's their blueprint of life. So we take a blood sample, we can understand what the genetics of this individual are. So this is a sequencing test now. Now, your genetic sequence actually is around about 3 billion base pairs long. Okay, so that's a lot of sequence to go through. And often what we're doing is scanning through and just trying to find maybe even one of those base pairs, one of those little bits of the DNA sequence which is causing the problem. It can be just a single base pair out of the 3 billion. It's kind of like playing a big game of Where's Wally? There's 7 billion people in the world, right? And you're going around trying to find Osama bin Laden, the bad egg, to hopefully revert the health. Okay, so these 3 billion base pairs are actually kind of condensed into the functional units of the genome, which we call genes. And we have 22,000 genes, and I'll get to what a gene is in a minute as I explain a little bit deeper. But needless to say, reading this DNA sequence is incredibly difficult. And in fact, if you look at the very first genome, which just had its 21st birthday party, this is the first genome published in 2003 in a landmark study. It actually took 13 years to sequence the 3 billion base pairs of just one person. Just one person. It took $3 billion, right? This is not something that you do in a diagnostic setting. So one person, 13 years, $3 billion. Now you fast forward to today, and we're living in a new era of reading DNA. New massively parallel sequencing technology, they call it. Today, you can get a genome done in less than a day, and it costs now, if you ask the right vendor, around about $100. And it's at the stage now where we're at it. We have programs in Australia, things like acute care genomics, right? Where a newborn infant in the first couple of days of life can be found to be really struggling. And they can do a DNA test, a sequence, and have the results, not only read the DNA, interpret the DNA, try and find that broken piece of DNA and return the results back to the clinician in less than 24 hours so that they can make real life and death important decisions based on that genetic diagnosis during the first days of this individual's life. That window of opportunity is critical for these kids, okay? So it can be quite rapid. Now, most times, if you came here in Australia, and actually Adelaide is very famous for this. It's the first place in Adelaide, let's say pathology, where a whole exome sequencing test was NADA accredited. It's now also on the Medicare benefit scheme, so you can your clinical geneticists can order a test, they can sequence your genome and hopefully give you back your result. But this can take months. This typically does, and that's not because of the need or want by the people involved. It's just the limitations of the workforce. So the technology of being able to create sequencing data has not been at this current stage met by the needs of the workforce to upskill and interpret these data at the same time. But it is possible, like I mentioned. In certain programs, we can get a result in less than 1 day, but typically this will take months. Now, because of the price of sequencing, because it's so cheap, now there's millions of people worldwide that have had their DNA sequenced. And this is really important that we aggregate this information together because it can tell us, the aggregation of healthy individuals' genomes can really tell us what healthy, normal diversification looks like. Our DNA is all different from one person to the next to the next. That's what makes us individuals. I've got thousands of different changes in my DNA compared to Hayley, compared to Emmy, compared to any one of you in the room. The problem is working out what's healthy variation and what's unhealthy variation. And the only way you can really do that is to really aggregate a lot of healthy data together. So it's really interesting, the millions and millions of DNA sequencing that we pull together, the more we'll get an understanding about health and also disease. So, as I mentioned, finding that genetic diagnosis, we can relay this information back to the family that can provide them the appropriate care, perhaps access to therapies, certainly family planning, and in rare diseases, access to a community where they can feel like they're not alone. Okay, so this is an interesting concept for you to think about. So we can all get a genetic test now, okay? But when's the right time, and do we want one, and who should have one? So here's the world's shortest person standing next to the world's tallest person at the time this photo was taken. And they can get a genetic test and say, yeah, this is why you're short and this is why you're tall, okay? That gives it, that's a diagnosis, okay? No problems, but can you act on it? Well, no. What you'd rather have is something with prognostic value. And this is why, particularly in neurodevelopmental disorders and other developmental disorders, you want to do a test really early because you want to be able to forecast what might happen to this individual based on that genetic finding, and then intercept with a therapy, with a care early on, so you can change the trajectory of life, so that this little kid doesn't have to imagine being either the tall or the short. You can make interventions that really help them be, or maybe they want to be tall. It wouldn't be too bad. Not that short people are bad either. Just want to say that as well. Good. We all have our place. So yeah, the timing of the test and when we can intervene is also a really important, thing to think about. So why don't we just sequence every kid? Wouldn't that be cool? Could we have, in 30 years' time, could we just walk into the clinic and go, well, my heart rate, well, my blood pressure's been high, to the GP, and he goes, well, have you got your genome sequenced? Maybe I can have a look at what's underpinning that health. And so, yeah, I mean, in a way, it's a personal thing, because you might also discover the underpinnings of your health, but maybe not all of them you want to know. What happens if I'm 2 years old and mum and dad sequence me and they find out that I'm going to get a late onset dementia like Huntington's disease, which I don't necessarily need to worry about until I'm 50 or 60. But perhaps if they tell me when I'm 12, I have to grow up my whole life with that burden of knowing that sooner or later that's going to happen to me. Maybe that means I won't have my own children. Or maybe it means that I've got the choice not to have the children because I don't want them to go through the same burden. So you can think about it. I mean, we often do this. How many people in the room would have their whole genome sequenced? How many would like to know what's going to happen to them, or how much of their life that they can predict based on a DNA test? I'm not sure. And it's definitely a new and evolving environment that we live in. How we use genetic data, how it implies what you think your health is going to be, maybe what an insurance company think your health is going to be. So we're moving definitely into untested waters. Certainly the technology is there to sequence a lot, but I guess we need the legislation and the governance around who should be tested, how tests should be done, and how results should be returned. So it's a really interesting environment and changing landscape that we're living in. So how is the information used? Well, certainly we're using it very well to discover new causes of rare genetic diseases. Since this technology came into, there's been an exponential increase in the number of gene diagnoses and number of known genes involved in these disorders. Precision medicines based on these genetic findings are evolving, and as I mentioned, there are thousands of clinical trials that are using this genetic information to try and build these great medicines, like this one, Spinraza. Spinraza is a new medicine. At the time it was developed about 5-10 years ago. It was the most expensive medicine ever made, right? But this is a wonder drug. These kids, you treat it for children with spinal muscular atrophy, right? This is a muscular degenerative disorder. These kids, they're born, they do okay, but then they start to regress. They end up not being able to walk, and they eventually you can't use their muscles to breathe. They're giving this drug to these children now, and it completely restores their health, and they can expect to go on and live a life as long as the rest of us They just need 3 injections or 4 injections per year of this drug. It's remarkable, and there are drugs based on this same technology that are out there to evolving to treat many other lethal or highly debilitating genetic disorders. We can also use it to work out how we will respond to medicines. So maybe later in life you might need you might have a leukemia or a cancer, and you might need some sort of chemotherapy. Now, we all, based on our genetics, metabolize these drugs differently. Okay, so the dose that your cancer and you would respond to, because you metabolize it differently, would be different to the dose that I would require because I metabolize it differently. Now, wouldn't it be great to go in there with your genome sequence and say, well, this is based on my gene sequence, this is how I metabolize this drug, so I'm going to increase the dose. So it's not just about developmental disorders, it's about all different aspects of health, how you respond to different treatments, different environments. So genetic causes of health are also important. There's a really cool study that was done a few years ago I think it's somewhere in New South Wales. It's called the Welderly study. This is where they took individuals that are 85 years older with absolutely no problems with their health. They're old and they're healthy. So this is quite an interesting cohort to sequence, right? Just like at the other end of the scale, we're sequencing individuals that have trouble with early health issues. If we can sequence people that survive all the way through to the later stages of life and still be completely healthy, there's some information in that. And as I mentioned before, even just aggregating data from healthy individuals gives us that idea of what healthy diversity looks like, and can teach us therefore about what a bad change in DNA might look like. And diversity of life, I guess it kind of touches on just what I said there, but they're also sequencing they're trying to sequence every organism in the world. The UK at the moment, through this thing called the Darwin Tree of Life, are trying to sequence 70,000 different organisms in Ireland and UK. But this is part of a larger project where they want to sequence pretty much every animal that is on the Earth. This is planetary health, so your health depends on the health of the planet. One of the key things about planetary health is diversity, and diversity is being drastically reduced since industrialisation taking over. So this is really important for our health, perhaps a little bit indirectly, but understanding the diversity of life and protecting that diversity is critical for the planetary health. Okay, good. So I've mentioned how cool our sequencing technology is, and currently we can get the whole sequence, and most of the time we'd hope we'd be able to find the DNA problem. But unfortunately, in 50% of the cases, even when they go through the most advanced clinical diagnostic pipeline, only about half the people actually do receive a genetic diagnosis. And there's a number of reasons that underpin that, and that's because sometimes we don't know what a deleterious change in the DNA might look like, but sometimes we need to go beyond the DNA to really understand how a change in DNA might impact the biology of the organism. And so in these instances, we just need to go beyond the DNA, and I'm going to explain what I mean in 2 minutes, to gather more evidence. So if we've got a variant, one that we suspect might be the cause of a disorder, but we need more evidence to sustain that. We call these variants, these DNA variants, variants of uncertain significance. Very suspicious, probably likely, but in a diagnostic setting, you need to be 100% certain, because as I mentioned, that might change the treatment you get, that might mean the clinical trial you're in, that might mean the family planning decision that you make or other family members make. So it needs to be 100% certain. But these variants of uncertain significance, a little bit more evidence can typically push them over into being certain. So what do I mean by going on the DNA? And this comes back to my description of the genes. I told you before, DNA, your chromosomes, encode 3 billion base pairs. That's split into 22,000 genes. 22,000 genes are the functional units of the genome. So what does a gene do? So this is the central dogma of molecular biology, that your DNA is split into these segments called genes. And what a gene does is essentially makes a protein, okay? And it does that by, first of all, taking a photocopy of a small part and making another molecule called RNA, which is essentially the same molecule, just with a slightly different chemical modification. But that RNA molecule can then be translated into a completely different type of molecule called a protein, which is a polypeptide made up of amino acids. Okay? And the proteins are the things that do things in cells, and we're all made up of cells. Okay? So here's an example of two immune cells, a B cell and a T cell, and I'm pretty sure you may not be able to read all that small writing, but all of these, just let me get my pointer. All of these different decorations on the surface of this cell are different proteins. You can see them listed down here. There's about 20 different proteins that are decorated on these cells. Okay, and these proteins give these cells their unique function. Okay, so the proteins that make an immune cell work will be different to the proteins that make a brain cell work, because these cells have different functions. The immune cells is going to try and find foreign pathogenic material and raise an immune response to protect the body from it. Whereas a neuron cell, its main role is to form connections that connect your brain to your muscles, or maybe empower you with high cognition and thought. Okay, so very different specialized cell types. Now, there's a set of genes which are unique to both cell types. We call them housekeeping genes, right? These are the things, like, I guess you could use the analogy that every cell is like a different sort of building. You have a bakery and you have your communication hub. So whilst they might all be made of bricks and have a front door and that, the inside they've got different specialised bits and pieces. So the housekeeping genes are the same, that's the bricks and mortar, but what's actually inside these buildings, making them do their different functions, is different. And these are the cells, these are the types of proteins that are commonly causing these neurodevelopmental disorders, are the proteins that are required for brain cell function. Okay? And so when we have a look at a variant which we suspect might be causing a neurodevelopmental disorder, we would think, well, this gene is probably going to be active inside a neuron cell. So it's going to be making proteins for the brain cells. So if we've got a variant of uncertain significance here on gene B, which is specific for brain, we can then maybe have a look downstream of it. So if we can't fully understand that it's deleterious impact at the level of the DNA, we might ask, does it affect the RNA, that little photocopy that's required to make the protein? Does it affect the protein? Or does it affect the way that the brain cell works? Okay? So that's when I say that we need to go beyond the DNA sequence to understand some of these variants. What I'm really talking about is that we need to have access to these gene products of RNA or protein, or even the cell type, to really understand whether these gene variants culminate in a defect down the chain a little bit. Now, these things that affect RNA or protein are often very difficult to tell based just on the DNA sequence alone. And that's why we need to go a little bit further. And that's been a hot area of research for our lab over the last few years. Okay, so it really comes down to it then, for some of these variants for which we would like the RNA or these products of gene expression, that we really need to get our hands on the brains of the affected individuals, because that's where these products are going to be found. But no clinician in their right mind is going to take a brain biopsy for the purpose of a diagnosis. Okay, it's almost, I can't quite work out the term, but it's very risky. It doesn't serve the purpose of helping the patient. It's more likely to harm the patient for the sake of a diagnosis. So I spent a lot of time actually trying to solve this problem in the lab. That is, how do we get our hands on this material to study these variants in the absence of being able to get a sample of brain from the individuals? Okay, one way that this is done all around the world is to use an experimental model. So we don't necessarily use the patient, but we use cells in culture or maybe a small animal model, and we engineer these models to have these genetic defects in them, and then we can kind of assay their impact in a laboratory experimental type environment. And quite commonly, mouse is used. This is very early embryo. This is very early brain development of a human and a mouse. This is around about just a couple of weeks of gestation. Okay, and you can see the similarities between human and mouse. And this is very reminiscent of basically most mammalian developmental stages that very early on, these primordial events look very similar, independent on what species you are. They get obviously highly divergent, and I'll show you in a minute what that can look like. But needless to say, starting the very early stages, you can use a mouse reasonably faithfully to model what's going on in perhaps a human. We can also do this in another way, like these mouse embryos are inside the mother, they're very small, very difficult to work with experimentally, but you can take stem cells from these mice and essentially then grow these brains inside of a dish. That gives you a bit of a renewable resource as well, something that you can experiment, you can do over and over. So another model that we use is stem cell based brain in a dish models. Okay, and I'll show you a quick example. This is a set of families. When we discovered these, no one had ever suspected this particular gene called USP9X was involved in neurodevelopmental disorders. Yet we have these families where these boys, you can see the black squares, they're all boys, all had an intellectual disability. Okay, and when we sequenced them, amongst all the different variants we found that they in common, they had these variants in this gene USP9X. This is a single base pair change, these very rare things I was talking about before. A simple T to an A out of the 3 billion base pairs, that's just one switch, right? Or a C to an A in this family, or just a deletion of an A in this family. And when we looked at this, we had a few other pieces of evidence to suggest to us that this gene might be the smoking gun here, but we weren't too sure. It was a variance of uncertain significance. We needed more evidence. So one of the things we did was to set out and ask, well, is this one of these genes that is expressed in brain, right? Part of the puzzle. And we did this. We had a look, where is this gene expressed? And we can have a look at where its RNA is expressing by staining this embryo, and wherever the RNA is expressed, it goes purple. And so we can see quite clearly that this gene is expressed most highly in the developing brain. Okay, what happens if we manipulate the expression of this gene? Our idea, our hypothesis was that those patients, that those variants might actually hamper the function of the gene. So what happens if we turn the gene down, the expression down? Does it impact the way that the brain grows? Well, here's a picture of a brain in a dish. And these are from stem cells, and they're quite remarkable that these stem cells, when you give them the right sort of environment inside the dish, they almost self-assemble themselves into these brain-like structures called a neural tube. Okay, very early on in the development, our brain is just like our intestines. It's just a tube. Okay, and this is a cross-section across that tube. And what you're seeing here is the middle of the tube, which is kind of like your lumen. And these are the stem cells in red, which kind of radiate outwards like spokes on a bike. And in blue, these are the mature nerve cells that are just starting to be born. And this is what it should look like. This is kind of like what now is called like a brain organoid. But this architecture, this neural tube architecture, was completely demolished when we started to manipulate the expression of this one gene, USP9X. So that tells us that this gene is important for brain development. Okay, we went on to grow these cells a little bit longer, and just like during development, you start off with a whole heap of stem cells that grow your brain, and then at some point they produce all the neurons of your body, and that's what you're born with, left with for the rest of your life. Okay, and in here we're seeing now less red and more green. The green are the mature nerve cells, and you can see you're getting lots and lots of nerve cells in this brain in the dish culture, but very few in this one on the left, on the right hand side. Good. So then we decided, well, let's try this in a bit more of a sophisticated model. Let's reduce the expression of this gene inside a developing mouse brain. And when we did this, and here is a brain of a control. Essentially, this is sort of like the nose of the mouse. These two nodes here, those nerves head into the nose so it can receive its sense of smell. And this would be the back of the head here. So you've got essentially the left-hand side of the brain and the right-hand side of the brain. And wherever you see one of these kind of stripes is a neuronal cell connection, connecting different parts of the brain together. So we're just looking at all the neurons in this brain in one picture. And you can see when we reduce the amount of this gene that we're really depleting the amount of nerve cells, just like we kind of saw in those previous brain in a dish models. We're reducing the number of nerves, and in particular, this particular picture looks at the nerves that connect the left-hand side of the brain to the right-hand side of the brain, a part of the brain called the corpus callosum, which is really important for learning and memory. And lo and behold, when we start to then go back to the patients and ask what's their major sort of phenotype, when we can get a scan of their brain, this is sort of what it looks like. This is not a USP-INX patient, but this is what they look like. This is just an image I chose because I think it illustrates the picture that you can see these on the left-hand side, these red connections between the left-hand side and the right-hand side of the brain. These are completely absent in these patients with USP-INX. Now, we went on and did a lot more experiments to show that those particular base pairs gave exactly the same phenotypic outcome in those different models. And so using this combination of stem cells and mice, we were actually able to prove that these particular DNA changes were deleterious to the activity of the USP-INX gene. And if you have decreased activity of the USP-INX gene, you have a problem with brain development. So putting all the pieces together, we were actually able to make a genetic diagnosis after years and years of research into this project. So that was 3 variants in 3 families. As time goes on, you make this publication and then people start ringing you up because they start realizing that that particular gene, variants in that particular gene might be actually clinically important. And so we get about an email about once a month, maybe once a fortnight, with a different variant asking, what is your opinion? Do you think this is causative of my child's intellectual disability? This is not coming from parents, although sometimes it does. It's mainly coming from people in clinical diagnostics or clinical geneticists who are dealing with a child and they're desperate to give a diagnosis for the reasons that I talked about before. The benefits of a diagnosis cannot be overstated. So what do? Well, we start to collect all of them, we keep researching them, we try and resolve as many as possible. At this point in time, we now know of 120 families worldwide that have the variants in this gene. And as the numbers build, then we can also begin to build the new clinical phenotypes, well, the clinical aspects of the syndrome, right? Because this gene has quite its own unique set of hallmarks. Using very large numbers of patients now, this is more in the 40s or 50s, we can see that these kids all have problems with intellectual disability, speech and movement disorders, hypertonia. Some of them have seizures. A lot of them have this agenesis of the corpus callosum, which was that neuronal defect I just showed you before. But we can also see here that the males, which are in green, and the females are also slightly different. This is very intriguing. So the males, essentially, males and females will all have this brain features, but it's only the females that also have problems with other parts of their body, short stature, scoliosis, some hip dysplasia, some pigment problems. So, but this is really important now. It gets to the point where with a really well-defined clinical set of hallmarks for the syndrome, that now the clinicians that are in the clinic can better match up the pieces of information. Okay, I've got now a variant in USP-INX, but I also have a child with that variant that has all the clinical hallmarks of what we now classify as USP-INX syndrome. And these bits and pieces add up together to enable us to make better and more frequent diagnoses. With that amount of people, another a great thing you can do is, like I mentioned, build a community. So 120 people worldwide. The parents are the experts of these children. We started a Facebook group just so that they can communicate together, okay? And it's quite amazing. Here's a quote from one of the parents after I led her into the group. She spent about a month communicating, reading through everyone else's posts, their experiences, learning from the others, and this is what she wrote. I honestly cannot explain what a difference this group has made to my life in just one day. It's been a very lonely 3 years, and at least I can have contact with people that understand. That's that power of bringing people together with rare diseases, that they are the experts. People are there to support them because they understand the challenges. We can also disseminate this information about the genes and their variants for access for clinicians and health professionals to access too. You can get this through adding your data in publications like we do, hosting Human Disease Genes website or the online inheritance man. All this information is available for clinicians so that when they have a variant and a matching clinical phenotype, they've got the information to help them put the pieces of the puzzle together to help make the diagnosis. Okay, now I said I was going to get to this. Mice are not mini humans. So whilst they may look very early on, in the very earliest stages of the embryonic development, you can see by the scale of things here that a mouse brain is orders of magnitude different in size and in complexity to a human brain. So whilst these mouse models that I just described have been super helpful in some genes, they definitely had their limitations. And there's been plenty of neurodevelopmental disorders that have been modelled in mice and then cured in mice that don't translate to curing humans. Right? Mice and humans, okay, they are different. So the idea then with the lab was, well, what other samples can we use? We should be using human samples. Can we get these human neurons? As I told you before, that would be what we'd really like, but clinicians rightly tell us we can't have them. Can we make neurons from other sources of the body? And it turns out that we can. So the accessible tissues, we've probably all had a blood test before. There's probably less of you that have had actually a skin biopsy. This is just where they take a small piece of skin under general anaesthetic, and you can actually grow the skin cells in the laboratory almost indefinitely, not completely. But these are the two sources that we looked at and thought, I wonder whether we could use either a blood cell type or a skin cell type to give us access to what might be going on inside the brain. And luckily there's some really smart people out in the science community, and we didn't have to invent this, but it's been a pleasure to apply it within the laboratory. We're talking about a Nobel Prize-winning discovery of cell reprogramming. So this was discovered in 2009 by Yoshi Yamanaka in Japan, and he worked out a way that you could take that skin cell sample, and with a little trick, which I won't go into the details, you can turn it into one of those stem cells that I was saying before that we could build brains from stem cells, right? So this is an embryonic type of stem cell. It's essentially turning back the clock. So you go from a cell from an adult and you kind of wind it back to a very early on embryonic state. And then from there, you can, using some other cell culture techniques, generate human neurons, right? So indirectly now we're taking the cell from a patient and we're now creating patient-derived neurons inside the laboratory. And you can see why this was awarded the Nobel Prize in such short time as well. I mean, some people have to wait for 40 years for a Nobel Prize after the discovery. This was awarded very quickly, and because it's had a very large impact on the way that we model human diseases using, it's basically giving us access to modeling different types of human organs. For my instance, brain, but it can be other cell types you can generate from these stem cells in the laboratory from the patient without taking a highly invasive biopsy. So a few people in my lab have played with this technology. We've modelled a few epilepsies, Sanfilippo disorder is another disorder we've modelled using this technology, not so much for diagnosis, but really trying to understand the pathology underpinning these neurodevelopmental disorders. And more recently, Olivia in the lab, who's with us today, had a really cool project last year to try and speed this up, because this whole process takes 6 months. And Olivia wondered whether she should shortcut it by going straight from a fibroblast to a neuron. It was quite an ambitious idea, but others in the scientific community had tried it. And when Olivia tried it, indeed she could. She took these fibroblasts and she turned them into these cells that express a lot of those neuronal cell specific genes and proteins, and they function like neurons in the lab as well. So now we've got 2 ways to really get access to patient-derived neurons without needing to take a brain biopsy. It's quite remarkable. This process that Olivia came up with only takes about 6 weeks, which means it's at a scale now that we could probably do it across larger numbers of patients for the purpose of diagnostics, for example. Patient neurons are, it's just an amazing resource. We can use it for aiding genetic diagnosis. We can use it to model these disorders, a lot of them occurring or arising during embryonic development or early development. We can model those stages. We can also use these neurons to do, drug screens, toxicity studies. There are other projects in Australia which are collecting these skin cells from patients with epilepsies which can't be treated with any of the known anti-epileptic drugs. So they're taken from the patient and they're able to grow them at large scale and do toxicity and drug screens to try and work out what drug might be best tailored for that person's genetic cause of epilepsy. That's precision medicine, where you go from a precise DNA change to a precise therapeutic. And that's the power, I guess, of doing genetics, but also marrying that up with a tailored option. Okay, so that's one way that we could access, I guess, these products of gene expression that are specific to the brain using patient materials. How else can we do it? Well, Emmy, who's also with us today, thought she'd take some inspiration and look at another Nobel Prize winning technology of CRISPR gene editing. And in this instance, she modified the system a little bit. So now she takes the skin cell, and rather than turning it into a brain cell, she just says, she points out the gene that she wants to study, and she can switch it on. So every gene, as I said, some genes are expressed in all cells, but other genes are expressed only in specific cells. But she tailors the switch that decides whether it's on or off, and she can manipulate that. So she can now turn on genes which would normally only be in brain, and she can turn them on in the patient skin fibroblasts. And again, this is really cool. This is a photo of the genes, and we know it's working because you get the green light, right? They turn on, they go green. That's when we know the machine is working, and we can actually get RNA and protein from these different genes. So here's a list of around about 20 genes. These are all genes which are only normally ever expressed in brain. And one at a time, Emmy's gone through and turned them on in the skin cell, and using that RNA from patients to help diagnose based on the way that the RNA molecule behaves. So all together, we've got this new kind of cool toolkit to really study genes that would normally only be accessible to study using a brain biopsy. Now we can use it by studying a skin biopsy, and that makes it very attractive in terms of diagnostics and actually rolling out to help individuals. So we had this study, it's called PERSIST. That's an acronym for a very fancy word, which I won't go into, but the idea is that with this study, we want to activate these disease genes using skin cells from patients so that we can resolve the pathogenicity of these variants. We're aiming to recruit 500 individuals. This study has been going for about a year now. This is a national-wide study. It's funded through the MRFF, and we are recruiting individuals from all over Australia with the hope that they can send us their skin samples of their individuals who have one of these variants which is super suspicious of causing the disease but need a little bit more evidence, and we're hoping to provide that evidence with our new technology of looking at brain genes in skin cells. Okay, so this is just the size of the team. I don't want to point out too much other than it's a very large project involving many people. This is clinicians, diagnostic labs, and researchers collaborating together across the whole healthcare spectrum, I guess you could say. But we also get support by the patient consumer groups like Rare Voices Australia. This is the national-wide voice for those individuals with rare diseases. Syndromes Without Names, again, it's another consortium of patient-driven promoters of trying to resolve individuals' genetic diagnosis. Epilepsy Foundation and Cerebral Palsy Alliance. So if you have any questions specifically about this study, you can email at persist@adelaide.edu.au, and we'll be happy to share a little bit more information with you. So I'm going to wrap it up there. I'm pretty sure I'm close to time. The biggest thanks of all of this research has to go to the patients and families, because we do this research for patients and families, but we can't do it without them. These precious samples of DNA or blood or skin that enable us to do our research are very, they're not taken for granted. This is it's particularly challenging when you're dealing with young children with neurodevelopmental disorders or intellectual disabilities or epilepsies. Asking them to take a needle can be quite challenging. So it's taken with great pride that these families are motivated to be involved in the study, and we treat their materials as super important to what we do. In terms of the research, this is just a list of some lab members, past and present. I mentioned Emmy just before. She's been working with the turning on genes in skin cells, and Olivia turning skin cells into brain cells. Claire Homan was also instrumental in setting that up in the past. We collaborate very heavily with another lab at the Neurogenetics Research Program headed by Joseph Getz. Joseph Getz is a very famous geneticist. He's discovered over 200 genes. I was once his postdoc, and I feel privileged to be in a great chain of researchers that have really looked at understanding the genetics of neurodevelopmental disorders. In fact, the very first gene involved in intellectual disability was discovered here in Adelaide by Grant Sutherland. The very first epilepsy gene ever discovered was discovered here in Adelaide by John Mulley. Joseph Getz pioneered discovery of genes mainly on the X chromosome, and as I mentioned, has discovered over 200 of them. And so I feel privileged to be part of that chain, and I work very closely with Joseph's group to this day. Also like to say that the Lister Lab's been important. This is a Western Australian laboratory who's been very instrumental in us picking up these new Nobel Prize technologies and implementing them in a new way, and also a long time interest in that one particular gene which I described, USP9X, which stems from the days of my PhD with Stephen Wood, originally here at the University of Adelaide, but now at Griffith, as well as some other collaborators in Queensland and in Chicago. And with that, I'll take a break and let you gather your thoughts, and I'm happy to take any questions. Thank you.
Thanks so much for that. That was super interesting, and you so clearly described some really complex topics there, and really, I guess, highlighted the importance in genetic research, not only for, I think, neurodevelopmental disorders, but I think health in general. So thank you for that. So we'll now open up to question time. For those of you who are listening online, you can submit your questions via the Q&A panel at the bottom of the screen. And then for those of you in the room, if you just pop your hand up, we have some roving mics coming around, so someone will bring you the mic to ask your question. So while I don't think we have any in the room just yet, but I've got a question here which was pre-submitted. So I'll ask that now just while you guys are warming up. So the pre-submitted question is, what tests are available to the public and are they covered by Medicare?
They are. So genetic sequencing tests can now be ordered under Medicare, and that's development as of about 3 or 4 years ago, and it was widely celebrated in our laboratory because we think it's important that people should have access to these tests, and it should be equitably available to everyone in Australia. So we're one of the only countries in which that is freely available, and there is a Medicare number for it. The test would have to come via your clinical genetic services. That's either at the Women's and Children's Hospital here in Adelaide or the Royal Adelaide Hospital. There are specialised teams dealing with genetics. And if you feel like you would like access to one, that would be the first point of call to go to. Now, recruitment into our study like PERSIST is dependent on that first genetic test result. So all the more advanced tests I talked about at the end of the talk today, which we do as research, first of all require that initial genetic test through a diagnostic laboratory. Yep.
Fantastic, thanks for that. Do we have any? There's just one up the back.
Thank you for sharing your research methodology. It's brilliant, and it was, for a layman, I think I walk away with some understanding, so thank you. My question is about, you mentioned how easy it is to get genetic tests done. My question is, who owns the information? Is that shared between pharmaceutical companies and research organisations, or is it controlled, and is there any methodology being put in place to try and open those doors so it is shared between you and the pharmaceutical companies?
Yep. The information is highly controlled, and it's considered personal information like any one of your other clinical records. So in no means when you get a genetic test done through a clinical genetic service is your information going to be distributed beyond those that are dealing with your immediate clinical care. So that's generally at a nuts and bolts sort of place, de-identified and stored as data, just ones and zeros, that could not be in any other way relayed back to the patient without the consent of the clinicians. So Clinicians can share information, like for example, in our study that we do, we first of all would need to consent the individuals to be involved to our study and explain to them that their information would be provided to us, and again, we are then the custodians of that and making sure that it is safe and not distributed to any other third parties. So it is valuable information. It really is. I could see insurance companies and drug companies would definitely like to have the aggregated information of people's genomes across different disease cohorts, and for that purpose is highly restricted. Yeah.
Oh, we have one at the back and then one down the front next.
Could CRISPR technology be used to reverse the deleterious effects of the variants in genes like USP9X?
Yeah, great question. And that's one of the promises of the technology that gene editing has really delivered, a very precise way of making gene changes, which could be, as you mentioned, reverting changes which are considered deleterious. Now, the first CRISPR therapy has indeed been FDA approved and is available in Europe. This is one where they're trying to revert retinosis pigmentosa, so a disorder with the eye. And that's a kind of a good test bed because to deliver the components to do the gene editing, you need distribution across a large number of cells. Now, the eye itself is a bit of a unique microenvironment where delivering these components, which typically happens by an engineered virus, can actually engineer enough cells in the eye to revert that enough to make vision. But there are a whole host of other disorders which are under development in terms of using CRISPR engineering to revert them. So it's definitely a watch this space. And there's a few pioneering studies that have already been approved, but there's a whole wealth of studies that are moving forward with trying to resolve all different types of genetic diseases in general. Now, specifically for USP9X, I think it'll be challenging because that's delivering to essentially every cell in the brain. We feel also that particular disorder might have its origins in early development. Like I mentioned, the neurons just don't grow and connect, okay, in the first instance. So it's almost like the time window for that disorder might be a treatment in utero. Can we fix that at a later time point? I'm less clear about, but certain genes, certain genetic disorders where the window of treatment is in adulthood or in early childhood, I think those disorders may be the best ones to target with a gene editing type approach.
Thank you for a presentation which obviously demonstrates a passion in your subject. My question is a more fundamental one, taking you back upstream. You mentioned earlier about where's Wally, and I take from what you're saying it's a bit about needles and haystacks. My question is, with your variances of uncertain origin and the like, how do you identify what's a variance and what's normal? Can you give me an easy layman's explanation of how you find your starting point, which is the gene you're focusing on in the first place?
Yeah, I guess you can filter down. Certainly if you've got a really strong clinical phenotype, that might reduce the number of genes you might want to look at. So instead of going the whole genome, you might just focus on, okay, I might just focus on the known epilepsy genes. And based on the phenotype, I'm pretty sure it's going to be one or two of these or three of these genes, because sometimes that genotype-phenotype correlation is that strong. The clinicians are that well skilled that they can almost smell the DNA change coming. And so you can order, you can order gene panels where you just go, I just want to look at these genes because I'm pretty sure it's going to be one of those. You could then go to something called the exome. So as I mentioned, the genome has 22,000 genes. Those 22,000 genes, the bits are really important to code the proteins, only make up 2% of the entire genome, right? So you can just say, well, I'm just going to look at the important 2% that encode genes, right? And if that still fails, then you're perhaps looking at the non-coding regions. And those non-coding regions, which don't encode proteins, they're all the switches that help turn genes on and off at the right time. And that can be very challenging, and that's perhaps where a lot of those unresolved cases will still remain. Even if we do get access to RNA and DNA, those sort of changes are more challenging. So you can take a stepwise approach if you have a strong hypothesis. But typically, this exosome, where you look at the 2% of the genome which codes genes, is a pretty robust of carrying all bases in a single test, and is relatively simple to look through. Now, I guess once you have all that information, you really do begin to focus on genes which make sense, those that have already been published in the past. So that's why when I say USP on X, we did a lot of research to show in the first instance that it was involved. But since then, now that we've kind of forged ahead and let people know this, changes in this gene can cause this disorder. Now when people look through that gene and they find changes, they become suspicious of it. Whereas in the past, they might not have. So the more genes that we understand are involved, the better the diagnostic rates become as well.
Thank you for the wonderful presentation tonight. It gives us a buzz to know it's all happening in the Adelaide University. A question to you. Some years ago, probably 10, my GP decided to try and work out an issue, and back came a genes test with the term a wild gene. What's a wild gene?
Yeah, it's a non-standard term, I would call it. No, sorry, I'd have to get a little bit more detail on that. Now, I guess that's the language that clinicians might use in a way to try and explain something in a lay terms. And I can only speculate that maybe wild means one that's perhaps not functioning as maybe he suspected it would do. Now, but I'd literally be hand waving. There's not a scientific or clinical term that I'm familiar with. I feel like that's a bit of a lay explanation.
In the interest of time, we've got one more question at the back, but we might make that our last question for tonight.
So I have probably a somewhat two-part question here. So when you mentioned through genes, we're able to perhaps assess what one might be more predisposed to, let's say. Are we able to, I suppose, assess for the level of severity for one disease that a gene might predispose, someone might be predisposed to? And second, I suppose, given that often, let's say, developmental disorders, particularly with intellectual ones, let's say as an example, there might be a spectrum on which someone is affected. So in cases where someone might have a mild disease, let's say, so you mentioned the benefits of diagnosis. I suppose on the other side of the coin, where there might be only a mild effect, would sticking a label, let's say, with autism or similar type diseases cause more harm than good for someone with only a mild issue where they've been diagnosed?
Yeah, that's a great question, actually. It makes me think a little bit. So, I mean, I guess the individuals that I'm dealing with were right at the severe end of the spectrum, and that's generally where my research lies. But yeah, we're on the spectrum of neurodiversity, and I did mention that, all of our children learn at different rates, different paces, and some people, a label might be beneficial. So it might be a conversation with a clinician. If you get a label from the clinician that diagnoses you with autism, does that give you benefits, gives you access to things at cost? Or do you want to carry that label? It can be, I guess, a personal choice. And things like, autism, I consider that not necessarily, there can be severe autism, but it's usually severe because of the co-symptoms within autism. Like it might be a comorbid with severe epilepsy or movement disorder. Whereas we all know, autism spectrum disorder can be actually quite mild, and these people can be quite high functioning, and they don't want a label. And it's really interesting to be involved with these consumer advocacy groups like Rare Voices Australia or Syndromes Without Names, because these are groups that are put together by parents of children that are dealing with these issues, or whether they want labels or whether they don't. So it's completely guided in a way where we tailor our testing, what cohorts that really want to be resolved, and those that want to be left alone and just left to live their life as best they can, because it's not necessarily a problem for those milder cases. It's just they just, I guess, need people to understand that they're a little bit different, and everyone's a little bit different, right? So so being on that spectrum is not necessarily a reason to to seek a test or to gain a label, but there might be some benefits if that's a personal decision. But we're generally dealing with the really severe end of the spectrum, with parents that are desperate for a solution. In the most severe cases, these children could have years to live, early childhood demise. And I deal with these individuals, and they're very desperate for diagnoses and precision medicines.
We'll take the time now to thank our fantastic speaker, Dr. Lachlan Jolly. And of course, thanks to all of you who are here tonight and for your fantastic questions. This event wouldn't be the same without you. So also be sure to sign up for our mailing list to receive the latest information on our Research Tuesday events. Thank you again all for attending, and I'll see you next time.